continue, we have now the next presentation by Niels Eijkelkamp, EnergiseME. Niels is managing the besides project also the youth and children biobank. And the microphone is yours.
It's important to keep the mic close.
So I'll try to give an update, 5-minute update of the project. And as Jos already said, this project has basically two main goals. One is building a pediatric biobank, and the other one is a more scientific one. So I'll try to cover both. Starting off with let's put everything on a bit busy slide, but I'll go through. Is building the pediatric cohort. This is also to highlight what we are currently doing. Also for those, please keep in mind, if possible, if you can take along pediatric samples, please do so. Here's also a bit of overview what's there. So the aim is to include 160 children aged between 8 and 18, and 80 controls. And that's all done with questionnaires and various tests, from assessment of the sensory system, doing the nasoline test, hand grip test, it's double, and we also do an echo for vascular stiffness. That was not shared. Another thing, yeah. I'll continue talking. So we're trying to include, so also with the patient organization, we have also tried to optimize the inclusion, so patients also have the possibility to participate in the study without going to the clinic. So really only for participating in this cohort. We have made amendments to that for the METC as well, that we can also advertise these now, the patient portals, and also to boost a little bit of the healthy controls, because the main thing that is tough to include healthy children in the cohort, and we need to advertise that more, and so we have to have approval for that, but that's arranged now. At the moment we have 50 children with CCC criteria and a bit less controls, 20, but we hope to boost that. The other cool thing, I think, what we could try do is also that we do a prospective study, so we follow these children for a year and also gain samples after a year. The reason for this is also what you have to realize. The samples we can take, blood, is much less for children than, of course, adults. So we're building a biobank, but if one takes a sample out, it's also very quickly empty. So we're trying to cover that. But this is kind of an overview on the right on the table. What are the samples? Just that you know, if you're interested in samples, we have PBMCs, but these are restricted to 1 to 2 vials. So what we try to do, if somebody is dying PBMCs, we try to align this with different groups so we can make most optimal use of these samples. And you see also the serum. I can share this later. Plasma EDTA, oral swabs, etc. So that's the pediatric cohort. So then the scientific question. So we're trying to build on the hypothesis, and you will also tell a bit more that autoimmunity, I think in the literature there's quite some evidence that autoimmunity may contribute to ME/CVS. And we're working on the hypothesis that autoantibodies that are present in the blood can activate the immune system, including monocytes, and that they can cause metabolic and mitochondrial defects in these cells. And that has consequences. We know that these cells, monocytes, this type of immune cells, can communicate with nervous cells, and that this disbalance in this communication leads to neurons not doing that well anymore. And that's what we want to measure by looking at altered responses of the nervous system, ultimately. So I'm trying to go through, how are we doing this? So the approach of our study is that we first, we're going to do a serum analysis and try to identify subgroups based on what's in the blood. So signatures of neuroinflammation, well, I have to see if that's really present. We're a bit early, of course, in children, and also other neuroinflammatory markers. We did some pilot studies. In January, we got the first samples also of the NMCB that we're going to measure. Then we're going to measure 190 adults, 50 children, and 20 disease controls. So that's going to be interesting. Well, we did some of a cohort that we had before. We found several factors, including a beta 42, which seems to correlate also with some of the symptoms. So that's the first approach, and then we want to continue with subgroups. Oh yeah, that's something I have to add on. The cool part is because Leo is now also involved, he will talk a bit more. Now, because the whole point with the biobank is we put stuff in the freezer, and a big cell population is lost, and that's the neutrophil. And well, if you talk with Leo about neutrophils, well, you see the passion about neutrophils, but sorry, Leo. But the cool thing is that we could now do point of care flow cytometry, so really measuring in flesh blood. So we can look at the cells that we thought were our favorites, the monocytes, but also neutrophils. And the real interesting part of the data that we have now is that in cell numbers, we do not see much on monocytes, but there seems to be something happening with neutrophils, and that really gained our interest. And that's a teaser for later for Leo to continue. But that's nice that we could do this on top. So collaboration is key and try to merge projects, I would say. Then I said metabolic disturbances. Also here we did some pilot work. And what I find very interesting and what seems to come out quite consistently also in adults, that several metabolic factors are affected, in this case in ME/CVS or pediatric ME/CVS. Also in QVS, we had this along as well. But as disease control, juvenile idiopathic arthritis, children with an inflammatory disease, no active inflammation, but a lot of fatigue, did not show those alterations. So we're now really diving into these factors, trying to identify if they're really causal to the disease, or is there a causal relationship. And then the other aspect is that we tried, as I said, linking the metabolism to function. Can autoimmunity affect monocyte function? And this is, we're not so far here, but we also developed a panel to measure specific autoantibodies. Carmen Scheiberbogen has shown this before, that autoantibodies like adrenergic receptors, which I think is also maybe relevant to the POTS project of Niels, but also that we can now detect these autoantibodies with assays that we have set up. And ultimately we want to also, this is really at the end of stage, combine all those factors that we identified, for example, these metabolic disturbances. If we mimic those, for example, in mice, do we get any symptoms? So really to show a causal relationship. With that, that's kind of the overview of the project where we are now, and I'm more than happy to take any questions.
Ik dacht misschien moet ik de microfoon nog even.
Keep forgetting.
Now I was wondering if you could tell a little bit something about the recruitment route outside of the UMCU. You mentioned 50 patients, at least children have been included thus far, according to CCC criteria. You could tell a little something about the progress there, because that's something we're working on together. But
yeah, so the most important part is that that we now have ethical approval to go through a different way. So one way is to go to the NMCB site and basically start the assignment to be participating in this cohort. And what will happen then, instead of being seen by a doctor, you have to acknowledge that you can also transfer your records of your doctor that's treated or the diagnosis. We try to get as much information as possible as the patient allows, and then you will be invited by the research nurse or Marta, who's not here today, she's on holiday, and will invite to come to the hospital to do the test, but you're not seen by the doctor, and you're only going through the tests as I just depicted, the POTS test, the pain test, or the pain test sounds very awful, so I can say that, but a sensory test that measure your sensory tests, and taking blood. So that the only part in the hospital is happening is blood draw, and that's it.
Yeah, thanks.
Yeah.
There are 2 questions online.
Yeah.
Sure. What are those disturbances that you were talking about? I don't remember the exact context.
The disturbances that we now see in the metabolism are linked to NAD metabolism. So redox, but I'm going really deep. But it's basically It says a bit of the potentially about the metabolic state. And the interesting part is that I'm coming from a chronic pain field where we saw similar disturbance in sensory neurons leading to altered neuronal functioning. So that's why we got very excited. And the other one is linked to LTB4. And that is particularly interesting because LTB4 is a very potent attractor of neutrophils. And now it's a little bit of a clock in the Claypole. But given that we see this increase in neutrophils and LTB4, we'll have to see if they are linked, these levels, because these are different cohorts that we measured. Else we could do associations between those, which we cannot do. So those are the two main ones that we have interest in now. I hope that helps a bit. There's a lot of it.
One more. Is it really necessary to use mice in your study?
We can have, I don't know how long. It's a cool question to do so. So now I think the most important, we don't only use mice, we use also human IPCs, for example, but I think it's very important. The point is we're trying to identify various factors that might be disturbed. But are there cause, consequence? We don't know. I think it's very important to design experiments, at least, to see if we can find causal relationships. And to give an example, for long COVID, together with Jeroen Dunne, we isolated, for example, these IgGs from patients and injected to mice and saw that they developed symptoms. And I think then these experiments become very important to really show that there could be a causal or a contribution of those factors to specific disease symptoms. And I think mice can help there, but of course, always be critical. What can you model in mice and what not, right? Because we cannot model all the symptoms. We can look at pain phenotypes. We can look at potential, well, PEM is going to be potentially tough, but we can let mice run and see if they have altered behavior afterwards. Is that PEM? Of course not. So you have to be a bit critical on this, but I think it can be very helpful to show the causal relationship between a factor and ultimate symptoms. And that's a short version of it, but we can have much longer discussions. Yeah.
That's
Yeah. Thanks. And I think time-wise, we also should